My name is Liz Worthey, and I am currently a faculty investigator and the Director of Software Development and Informatics at the HudsonAlpha Institute for Biotechnology. What I'm going to tell you about today is some work that we've done as one of the sequencing centers for the phase 1 of the NIH's Undiagnosed Disease Network. This is work that I did in collaboration with Howard Jacob, who was also at HudsonAlpha and is now moved to AbbVie. The UDN is really a program that was designed in order to bring some expertise and technical capabilities to the field of diagnostics for individuals with rare, undiagnosed or misdiagnosed disease. What this shows you here is a map of all of the sites, the phase 1 sites for the UDN. The setup for that was that there were a number of clinical sites where the patients would be seen. There were then 2 different sequencing cores. One was a whole genome core and one a whole exome core, one at Baylor and one at HudsonAlpha. And then later there were a number of additional sites that were brought on board, including animal models and metabolomics. And all of that was organized through a coordinating center that was at Harvard. As I mentioned, the focus of the UDN is rare disease. The international definitions of rare disease vary. In the US, it's a disease that affects fewer than 200,000 individuals. Unfortunately, 30% of children with a rare disease won't live to see their 5th birthday. There's actually more than 7,500 different types of rare diseases, and the majority of them are believed to be genetic in nature. 10% of the population has a rare disease or a rare form of a disease. So you can see, although the diseases are rare individually, rare disease patients are not rare themselves. The average time for diagnosis for an individual with a rare disease is more than 7 years, with at least, on average, 8 attempts at a diagnosis. Normally, individuals get 3 or 4 misdiagnoses. 35% of the deaths that occur in children in their first year is due to the presence of a rare disease. Clearly, that's a huge impact to those individuals and their families when they get one of those diagnoses. But it's also a huge impact to the healthcare systems that treat these individuals. And having a rare disease often affects all aspects of care for those individuals. An individual with a rare disease, even for their normal follow-up or primary care visits, require longer, and then they require more of those visits. More than 60% of individuals with a rare disease have received highly conflicting treatment suggestions. 95% of rare diseases don't have an FDA-approved drug treatment. There's only a couple of dozen centers of excellence for the treatment of or diagnosis of individuals with rare disease through the application of genomic medicine, which I'll come back to. It's estimated that more than 10% of the inpatient costs in a hospital system come from the 2% of rare disease patients. And overall, it's more than 6.5 times more expensive to treat those patients that have a rare disease. The talk today is going to focus on molecular diagnostics. So how do we obtain a diagnosis, a definitive diagnosis for patients? Back in 2003, my clinical colleagues told me that if you were unfortunate enough to have a child who was diagnosed with a rare disease, and you were lucky enough to live in the United States, and you were lucky enough to live somewhere close to one of those centers of excellence, and you were lucky enough that your child was well enough to get to one of those centers, and you were lucky enough that your child didn't die shortly after birth. If you went to one of these clinics, on average, about 5% of the time, you would leave with a definitive molecular diagnosis. Most of the time, you would not have that. Between 2003 and 2010, the world changed a great deal. One of the things that was important for my field is the advent of almost routine genomic sequencing of individuals. In along that same time, we had lots and lots of advances on the informatics or the computational side. By 2010, however, despite those advances, you would only leave that clinic about 5% of the time with a definitive molecular diagnosis. One of the things that we've seen since 2010, however, with the advent of what we call genomic or genomic precision medicine, is a dramatic increase in the diagnostic success rate. So in the 2012-2013 range, it had gone from 5% to the mid-20s up to the mid-30%. In 2015, it was up at 43% perhaps, and arguably currently that diagnostic success rate is in the 40s, high 40s or 50s. So what do I mean by genomic medicine? Well, this is the process where we have a patient, we extract DNA, we obtain blood, we extract DNA, we go on to sequence that DNA using one of these next generation sequencing machines. We then take those short sequence reads, we map them to the human reference genome, we identify all of the places where that patient is different from that reference. We put that into some tools that allow us to analyze that data. At the same time, we obtain the patient's clinical information, their phenotype. That goes into that same tool as well. And what we end up having is the ability to interpret that genome and derive from it a clinical report that then can be returned to the ordering physician who ordered that report. I wanted next to tell you a little bit about the cohort for the Undiagnosed Disease Network. HudsonAlpha, as I mentioned, was the whole genome sequencing core for the UDN. Within that, we saw 515 of these rare disease cases. In total, 577 probands. Many families had more than one individual who was affected. We saw, in addition, in total, 647 affected individuals. So in some cases, those individuals were not regarded as probands because they didn't go through the full testing. However, if we did make a diagnosis, we would also be able to give information back for those individuals who weren't full probands as part of the UDN. In general, the analysis was a trio. So we had the patient and then both of their parents. So we also had more than 1,000 unaffected individuals. For each patient, what we received was a referral letter and a set of records that had come from the clinical site. In addition, we also received a blood sample. And the process that we went through here at HudsonAlpha was to sequence, analyze, interpret, and return a report. This just shows you the primary symptom categorization for the patients in the UDN. As you can see, the text is small, but what it shows you is that we were seeing patients with lots of different types of disorders. The largest section there is patients who had neurologic symptoms, but we also saw all other sorts of rare disease patients. It's also interesting to look at the age of our patients. Again, what's important here is that on the left-hand side, you can see from the age newborn, so from 0, all the way through to the oldest patient that we had within the UDN was 89 years old. And we saw lots of patients who were adolescents, middle-aged, or actually older than that. 85% of those patients, however, had had that rare disease since childhood. So they may have been adults, they may have been middle-aged, but they had been aware of their rare disease for most of their life. Another interesting thing to consider is the drugs that these patients take. So this shows you, just from the clinical records that we obtained from those individuals, the count of the number of drugs that patients were taking. So all of the patients were taking at least one drug. One of the things that was a little surprising is that almost 60% of our patients take more than 4 drugs on a routine basis. Well, more than a third of them take 10 drugs on a routine basis. And it's interesting to think that not all of those drug combinations will have been studied together. And actually, few of them, or maybe none, will have been studied in patients that had the same rare disease and physiology as our UDN patients. So one thing to consider moving forward is the effect of polydrug interactions on patients with rare disease. Another thing that we looked at was the prior sorts of molecular testing in our cohort. And this just shows you a graph of a count of patients and the number of prior molecular diagnostic tests they had had. Everyone had at least one prior test, but even with the limited records that we have, we can see that 44% of those individuals had had more than 3 attempts with no diagnosis, and a quarter of them had had 5 attempts with no diagnosis. And we also had patients that had dozens of attempts with no diagnosis. As I mentioned, we were the whole genome sequencing site, so one thing that we wanted to look at in particular was, did these patients have prior exome sequencing? And we could see that 93% of them had. 40 had had no testing as far as we could tell from the records, other than the testing that they received within the UDN, which would have been either genomes or exomes. 76% of our patients had had a prior exome. And most of these were recent exomes, relatively high quality. One thing that we also consider is our solve rate. So normally when we're thinking about molecular diagnostics, we're thinking about whether we're going to return a finding from the molecular diagnostic lab that we think is an answer for that patient's clinical presentation. However, that's not the same as a solve in this case. Here we're defining a solve as an instance where something is returned to the clinical site and they agree that it's the definitive or likely diagnosis. So as I mentioned, that's a little different from the normally quoted molecular diagnostic success rate, which can include things that get returned to the ordering physician where they end up not agreeing that's actually the cause of the disease. So within our cohort for the UDN, we had 25% of cases were solved. And 26% of our total probands were solved. Interestingly, 6 of those solved probands actually had more than one disease. They had at least 2 distinct disorders. When we think about solve classification, not all of those solves came from the molecular diagnostic testing that we did. In some cases, all it took was for that patient to be seen by the diagnostic team at one of those sites. In addition, in some cases, the clinical site actually received the sequence data and they solved it prior to receiving a report from HudsonAlpha. So one of the questions that we asked is, in those cases where the patients had had a prior whole exome, which is the current standard, if you like, for an attempt at molecular diagnosis for a rare disease patient, why did they not get a diagnosis? In some cases, it was because the whole exome sequencing had been done a while ago, before that connection between the locus or the gene that the variant was found in and the disease had been made. That was the minority of cases. The majority of those whole exomes had been since 2016. Another reason is, of course, that the whole genome sequence actually gives higher, more consistent coverage of an exome due to fluctuations in sequence coverage. So the whole exome sequence data from the UDN covered 92.5% of the targeted regions at greater than 10x, and our 40x genomes actually covered 99% of those exome targeted regions. So you get better exome coverage from a genome. You can also obviously test much more of the genome when you do a whole genome and not an exome. And interestingly, when we went back and we looked in ClinVar, about 5% of known causal variants in ClinVar are covered by genome, but not by exome sequencing. And then when you do a genome, you can also identify other types of molecular variation that's difficult to identify with an exome. This includes small or large structural variants, for instance, structural variants that span an entire gene. It also includes other types of variants, like repeat expansions, which are difficult with an exome, and then larger chromosomal abnormalities, such as mixoploidy, which I'll come back to later. And again, as I mentioned, that coverage is important. It's also important because the more consistent the coverage, the more accurate you can call variants. That's something else that we noted. So I wanted to give some examples of the patients that we've seen through the UDN. It's important to say that these are the most difficult cases, perhaps, that have currently been put together as a cohort. As I mentioned, often they've already had extensive testing and they've already been seen by excellent geneticists and diagnosticians. So this is an example. This is a case from the NIH clinical site, their UDP. The patient had developmental delay, epilepsy, rigidity, bradykinesia, a Parkinsonian tremor, macular degeneration, retinal dysfunction, and skeletal abnormalities. And when we did our whole genome sequencing, we did find a pathogenic homozygous variant in a gene called PARC7, which is associated with early onset Parkinson's disease. Because this is a genome, we hadn't just tested that gene, we were able to find a second pathogenic homozygous variant in a gene called ABCA4. This gene is associated with macular dystrophy and retinitis pigmentosa. When we kept looking through that genomic data, we also found a third variant in LBR. This is an intronic variant. That gene is associated with Greenberg skeletal dysplasia. So what we see is that this individual patient had 3 distinct different diseases that were all contributing to their phenotype. It was a little suspicious that they were all on chromosome 1, and it turned out indeed that this patient had chromosome 1 paternal isodisomic uniparental disomy. If we had just been looking for maybe something associated with the Parkinson's, we might only have looked at PARC7, we may have stopped. So it was important that we did a whole genome so that we could find all of those different components of that individual's disease. It turned out, as I mentioned, that 0.5% of our patients have 2 or more disorders. We developed a pipeline to also be able to call structural variation, as I mentioned, and 20 of our solves came from this. About 4% of the solves had large structural variants. 6 were de novo heterozygous variants. 6 were reported as compound hets. 5 of those, it was a large structural variant together with a small variant. But in one case, we actually had, the individual had 2 structural variants inherited in the same gene. In 2 cases, we had homozygous inherited variants. In one, this was because it was inherited from a single parent, but it was homozygous due to uniparental isodisomy. Of note, in 4 of those cases, in the gene shown here, that patient had actually previously been tested with a gene-specific test, which had come back negative, which we then were able to show was not the case. And then we have many other structural variants that are, some of them are likely to be causal, they just need a little bit more work. As an example, this was an adult onset disorder case, a 40-year-old female, healthy until 32, initially reported with sensory neural hearing loss, then her eyesight and her memory and decision-making started to fail. She had sensory and motor neuropathy, and then she developed severe weakness in lower and bulbar muscles, and she was confined mostly to a wheelchair or a walker. Her symptoms got worse over time. They were much more severely getting worse at the start, and then they tailed off a little bit. She'd seen lots of different specialists, been to many different hospitals. This just shows a variety of the sorts of interventions that she had already been through before she came to the UDN. Lots of prior genetic workup. We performed a whole genome sequencing on her, and what we found is 2 rare variants in trans in the SLC52A3 riboflavin transporter, which means that she had riboflavin transporter deficiency. And then she also had a 3rd rare deleterious variant in acyl-CoDehydrogenase. And those 2 different genes that I mentioned are actually both parts of that same pathway. So this gave us a diagnosis. And this is the disorder. So it's Riboflavin Transporter 2 deficiency, onset between infancy and 30s, progressive disorder that presents with hearing loss, often leads to death due to respiratory failure. It's known that high doses of riboflavin often relieve symptoms. And one of the things that was interesting here was that this individual had actually received a mito cocktail, which is a mixture of different compounded drugs, because she had had respiratory failure a few years into her disease. And that has a low dose of riboflavin. So that's probably what made the severity of her symptoms tail off after a few years when she was put on this. With the diagnosis that she had, they actually increased the dose of riboflavin by 7 times. And one of the great things that happened was, whereas previously she had no sensation in her feet, afterwards, within 48 days, she had sensation returning and increasing improvements over the next few weeks and months. And before the treatment, she took a very long time to cover a short distance, and that time improved and improved dramatically. There's general agreement that the response tends to be better and faster with earlier treatment. So this was an example of a case where it doesn't happen very often, but the diagnosis led to a treatment that helped significantly improve that patient's outcome. There are other types of variation, obviously, and we've developed methods to identify them. This is just an example of a case from the Vanderbilt. It was a 31-month-old male with a heart defect, hypotonia, dysmorphic features, and some other symptoms as shown here. Able to sit without assistance at 7 months, standing at 24 months, but not walking. Few words and signs to communicate. He did have hyperpigmentation of the abdomen and legs that followed the lines of Blaschko. No family history, and only child. Lots of tests, lots of consults in clinics over 2.5 years, and no diagnosis. We performed whole genome sequencing on 2 skin samples from the light and dark skin, and our standard methods didn't find anything. But because of that hyperpigmentation, we had started to develop a pipeline that would help us look for chimerisms or mosaicism. Most people are diploid, 1 set of chromosomes from mum and 1 from dad. And when we look at the test that we developed, a normal individual who's diploid would have bands at 0, 50, and 100. But what we saw in this individual was some extra bands at 90 and 15 in the lighter skin sample, and 95 and 5 in the darker. And what that means is this individual is intermediate between diploid and triploid. So he has some diploid cells and some triploid. He's XY, XXY. This was confirmed. And we now know that he was a mixoploid individual. And we were actually able to identify another mixoploid individual as part of the UDN. So findings to date, we have made diagnosis with small variants, repeat expansions, structural variants, including uniparental disomy, mixoploidy cases, other types of chromosomal abnormalities. And the diagnoses are being made in many different categories. So we have known and recognized symptoms that just weren't previously caught. We've also seen expansion of phenotypes for some syndromes, so it didn't quite look like what it actually was, so maybe they weren't given the right sort of test, but a genome can pick that up in an unbiased manner. But then we've seen novel phenotypes for a known gene and new genes being identified. So working together with the clinical colleagues across the whole of the UDN, a total of 32 new syndromes were defined. In terms of actionability of findings within the UDN, 84% were medically actionable by the clinical teams based on defined and agreed network criteria. Many of these were specific changes in therapy, changes in the management other than specific therapy, such as narrowing of diagnostic testing, and then variant-specific genetic counseling. So I would say that we are doing much better. So as I mentioned back in 2003, that success rate was only 5%. And now the diagnostic success rate is 50% for patients with rare disease. However, even if you can identify the molecular variation that's causing disease in an individual, and you can interpret it and give a definitive diagnosis, you still have a lot of work to do to really be able to help that individual and their family and the individuals who are taking care of them. Because those people have 5 main questions. So what's the diagnosis? How did it happen? Who else is at risk? What can be done? And what can we expect? And in order to deliver an accurate answer for the last 2 questions, we need to understand some other things. So we have to better understand the factors that can alter a patient's clinical presentation. You can have 2 individuals who have cystic fibrosis, who have very different disease outcomes, who have very different progression and different symptoms, even when they have exactly the same CFTR variant. So one of the things that we absolutely need to be able to do in order to say what can we expect is to understand the rest of that individual's genome to see if we can identify factors that are modifying their phenotype. And then we also have to be able to understand how environmental factors can work with those genetic factors to change that clinical presentation. And the second thing that we have to be able to do is to understand the pathways that are involved and how those molecular variations are actually altering those cellular processes and pathways. With that information, you can actually start to work with industry partners or others who are interested in the development of therapeutics. And that's the second thing that you have to be able to do. If you really want to be able to tell a parent or a patient, this is the course for your disease, and here's how we can change it. And these are 2 things that we and many other folks in this field are now starting to turn their attention to. So hopefully that was a good summary for some of the things that we've been working on within the Undiagnosed Disease Network. And with that, I would just like to say thanks. This is lots of names, obviously. This is a lot of work, and there are many different individuals who contributed and collaborated, not least of all the patients and the physicians who were part of the Undiagnosed Disease Network. And with that, I will say thank you, and I will let you know that if you do have any questions, that you can submit them, and they will come through to my email, and I'd be only too happy to answer them for you. Thank you.